So there are many ways of looking at data, and when you set up your study, it's really important to be able to clearly articulate the purpose of the study, and then talk with your bioinformatician and statisticians about how you'll want to visualize the data at the end of the study. It's important to do that up front, and I'm going to spend the next few minutes describing a few of the possible choices with respect to how to visualize your data, and these aren't mutually exclusive, and in fact it can be informative to look at your data in many different ways. So this is just an overview of those possible ways of visualizing data. As I mentioned, the first step before beginning any project is to define the objectives. Many biomarker discovery studies can be categorized as class comparison studies. These types of studies aim to identify microbial taxa that change abundance in samples representing two or more groups, such as different treatments, locations, or time points. For example, a class comparison study aim may be to identify microbial taxa that are more or less abundant in the stomachs of patients treated with a drug. Samples are chosen representing groups with and without treatment, and the microbial profiles are analyzed for taxa that change in response to treatment. Class comparison relies on the assignment of a set of observations into subsets or clusters so that observations in the same cluster are similar in some sense. There must be some attribute that is shared. Clustering is a method of unsupervised learning and a common technique for statistical data analysis. A similar method is principal component analysis. This procedure transforms a number of possibly correlated variables into a number of uncorrelated variables called principal components, and basically challenges the relatedness of groups and can reveal unexpected similarities and differences. Another type of study is a class prediction study. Class prediction studies aim to assign an unknown sample to a class based on established signatures or predetermined microbial community profiles. Class prediction is very useful for assessing the power of the stratification or subclassification signatures developed in a pilot study, and ultimately for assessing the performance of a diagnostic assay. Ultimately, the practical application of the test that you will be developing is in the clinic, and it fits this model. Clinic applications fit class prediction models. Think about it this way. An individual's microbial profile would be matched against a database of well-characterized profiles with demonstrated clinical utility. When the patient walks into the clinic, they're not walking in with a cohort of 1,000 people that will be subclassified. You're going to take that patient sample and you're going to bounce it against the class prediction model that's been built as part of the clinical utility of the assay. So this model is a very nice one to run early to see how robust your signature would be for an individual sample. A third example is class discovery studies, which aim to uncover subsets of samples with distinct microbial profiles that may indicate a change in an environmental or human factor. For example, the aim of a class discovery study may be to identify a subgroup of gastrointestinal disease patients with a different underlying etiology. This example shows a heat map, a figure familiar to many of you acquainted with expression analysis signatures. Heat maps are a very nice way to visualize specimen subclasses and the taxa driving those subclassification signatures. So those are just a few examples. There's many ways to test your, to first of all, uncover the biomarker panel, and then secondly, to test it. One very important consideration in setting up a pilot study, and this is probably the most challenging of all the questions that you're faced with, is how many samples do I need? This can be a difficult question to answer, and frankly, unless you know how big the differences or changes are in the sets you're comparing, it's difficult to know. If the changes are small, more samples are required to produce a robust signature. Large changes or differences require fewer samples. There are statistical methods for estimating a baseline number of specimens, including simple t-tests or ANOVA, which provide some information. As you build your signatures, you will revisit this question to determine how robust the signature is. It's essential to structure your pilot study so that there are multiple specimens representing each type of sample, group, and time point. 5 in each group is a bare minimum. And although it may reveal a trend or directionality, this number is unlikely to provide statistically significant results when you're doing whole microbiome studies or whole genome studies. As you add specimens, taxa driving the signature, those that reflect the biology underlying the condition, will persist, while those that may be present by chance will fall away. And that's really the basic concept. When you're looking at a whole genome, you're going to find similarities within groups and between groups by chance, because you're looking at so many different aspects of the microbiome or attributes. So as you build your data set, you will be then smoothing out those things that are occurring by chance, and the aspects of the groups that are there due to the biology or the question that you're asking, such as treatment response, those things will become obvious. But I just want to caution you that it's always important to talk to a statistician. It's always a good idea to discuss your project with a statistician if you have one at your institution. And if you don't hesitate to call us and talk to us about your study. We'd be happy to talk with you and help you work through things to consider when setting up your pilot. So one of the things that I like to talk to people about when they're doing a comparison study is, why you need a representation of the group that you're, of the different groups that you're comparing. And I talk about this as the essence of cowness. So what you see here is a pasture of cows. They all look pretty much the same. They're all from the same herd. They're eating the same food. They're out in the same sunshine. And you would assume that any one of these cows would represent all of the cows. In fact, what you'll find is that these cows, their gut microbiome will have variation. There will be variation due to the individual cow biology. These cows are all individuals and they've experienced their lives differently. There will be environmental differences, and although there will be certainly aspects of their microbiomes that are shared, if you would sample every one of these cows, you would also find differences between these cows. And so the point here is that you need to have in your sample set, within your groups that you're comparing, you need to have a good representation of the biological diversity within any group. So this is just an example that I've used that I think helps people understand why this is important. You want to understand the variation within the group so that you can better characterize variations between groups that you're comparing. And any of the differences that your samples share within groups will be characterized nicely the more samples that you have within the group. So it's just something to think about when you're doing your study design.